In this first lesson on membrane structure, we're going to be talking about phospholipids and the phospholipid bilayer. And here are your objectives. So the first thing to understand about the cell membrane, also called the plasma membrane, is that it's selectively permeable. It lets some things through and doesn't let other things through. And we'll talk about why that is over the next few lessons. Next thing to understand is that the cell membrane is a phospholipid bilayer. It's made up of two layers of phospholipids. Well, what is a phospholipid? Here's a schematic where this part is a phosphate, a functional group made of phosphorus and some oxygens, and this part is polar. This is glycerol, and then attached to the glycerol here, we have two fatty acids. And the fatty acid tails of a phospholipid are nonpolar. Now, the fact that we have polar and nonpolar parts has important implications. Let's talk about a few terms and then we will think about those implications. The term hydrophilic means water-loving. And something that's hydrophilic is going to seek out water. Things that are polar, like water is polar, are going to be hydrophilic. Remember that like dissolves like. So the polar phosphate head of a phospholipid is hydrophilic. It's going to try and seek out water. In contrast, hydrophobic means water-fearing, something that is going to try and get away from water. And that's going to be a consequence of it being nonpolar. Again, like dissolves like. Water is a polar solvent, so it won't dissolve nonpolar fatty acids, for example. The fatty acids, again, are hydrophobic. They're going to try and get away from water. When we have a molecule like this that has a hydrophilic part and a hydrophobic part, we call that amphipathic. It's something that has both hydrophilic parts and hydrophobic parts. Both hydrophilic parts and hydrophobic parts. That's amphipathic. So a phospholipid is an amphipathic molecule. Now, the cell membrane is a phospholipid bilayer. I'm going to simplify this diagram even further, and we're going to see this a lot. The phosphate and the glycerol we're going to draw just as a circle, and then the fatty acid tails we're going to draw like that. So that's our new schematic, even simpler than this one, for a phospholipid. Now, if I had a bunch of phospholipids and I were putting them in water, the hydrophilic heads would want to get near the water. This is the water here. But these hydrophobic tails are going to try and get away from water. And the only way they can do that is if they pair up with other phospholipids facing in the opposite direction. In this way, if there's water out here, the tails, the hydrophobic tails, can come together and get away from the water, excluding water from the interior. So now if you just continue that process, you have water, aqueous solution on the outside and on the inside, but here there's no water in the middle. You've just got these nonpolar fatty acids. So this is a phospholipid bilayer, and it's just the natural conformation that phospholipids are going to form in an aqueous solution. It's the lowest energy state where we get the parts that don't like water away from water and the parts that do like water towards. Now, phospholipids make up the main constituent of the membranes, but they're not the only part. So let's take a closer look. If we have phospholipids, again, the aqueous solution is out here. So phospholipids with their hydrophobic tails pointing to the inside. There are also proteins in the cell membrane, and those proteins can be in two places. They could be right in between the phospholipids. So if there are phospholipids on either side like this, there's a protein in the middle, and this is called an integral protein. A protein that is embedded right in the membrane. This is an integral protein. We could also have proteins that are just on the periphery, like this. This would be called a peripheral protein, and those are the two major classifications of membrane proteins, peripheral and integral. we can also have carbohydrates, often they are branching structures, coming off of proteins. And if there were a branching or if there were a carbohydrate coming off of this protein, that would also make it a glycoprotein, glyco meaning carbohydrate. So this peripheral protein now is a glycoprotein. And we could have carbohydrates coming off of lipids as well. That would make them glycolipids. And these carbohydrate groups, like we see here, are important for distinguishing cells from each other. The ABO blood groups that you have probably heard about have to do with glycoproteins on red blood cell membranes. Also, in animal cells, interspersed between the phospholipids, we can have a steroid structure known as cholesterol. It's got 3 hexagonal rings, which I'm not going to be able to draw well here, and a pentagonal ring. This is cholesterol, and we'll talk more about cholesterol more in the next lesson, but it's involved in maintaining fluidity, especially at extremes of temperature. So that's a quick schematic of the phospholipid bilayer, and this cell membrane with the phospholipid bilayer as well as the proteins and carbohydrates and cholesterol is referred to as the fluid mosaic model of the cell membrane. The mosaic part refers to the fact that it's got all these different constituents and will have different composition in different parts of the cell. You'll see different proteins in different parts of the cell, etc. It's also known as the Singer-Nicholson model of the cell membrane after the 2 people who first developed it. Okay, so that's an introduction to membrane structure.